Hi, and welcome to the Anti-Waffle podcast of Em and Pixie.
Woohoo!
And together we'll try to talk about research without any gobbledygook. Each episode we'll be talking to a researcher who will discuss the work they're doing, looking at why this research is important and asking how did they get into this field. We hope that it will be informative and interesting and bring a bit of the academic world into the real world. Today we have Dr. Nathan Woodling from the Institute of Healthy Aging at UCL. Hi.
Hello.
Hello, thank you very much for being here with us.
Oh, thank you.
Thank you. Would you like to begin by telling us about your research, please?
Sure. Yeah, so just broadly, I work on aging and neurodegenerative diseases. So these are diseases like Alzheimer's, Parkinson's, motor neuron disease. We all know intuitively that these are diseases that increase with age. Older people are more likely to get these diseases. But what we don't really know is why. What is it about the aging brain that actually makes people get these diseases? And that's broadly what I'm interested in, figuring out why it is that the older brain is susceptible to these diseases. And my work particularly is on Alzheimer's disease, but I think it would be applicable to a lot of the different diseases as well.
Have you got any ideas of what's kind of causing it, any particular areas that you're looking at?
Yeah, I think we have two hints so far. One of which has been done by researchers here at UCL, where they've looked at what actually changes in the aging brain, looking at samples from people at different ages who've died at different ages to see what actually changes in the brain. And broadly speaking, looking at the brain, it's made up of a lot of different cells. We're probably all familiar with neurons. These are the cells in the brain that do the firing and the wiring. But surrounding all of those cells are also all of these cells called glia. And glia are the support cells, but they also do really important roles in regulating how neurons work.
Right.
And it turns out if you look just at genetic changes in the brain with age, it's the glial cells that actually change the most with age, whereas the neurons stay relatively stable. So that's the first hint.
That makes sense.
Yeah. Well, I think so. But the second thing has been to look experimentally at what actually controls aging and longevity. And that's been work done here at the Institute of Healthy Aging at UCL. And here, a lot of different researchers have asked the question, is aging something that's completely immutable, or can it be changed? And it turns out, in research looking at short-lived animals, like nematode worms or fruit flies, where you can actually look at the whole lifespan really quickly, you can have changes in single genes that can lead to a longer, healthier lifespan in these animals.
Right.
And those same genes are also the ones that are, they seem to be anyway, different in people who end up living past 100. So the same genes that seem to be impacting longevity in worms and flies are also, we think, doing the same things in humans. So we've got a cell type, glia, and a whole bunch of different genes that we know impact aging. And my research so far has been to ask simply, what happens if we change those genes just in glia? And I've been doing that in fruit flies because they're really short-lived and I can look at their lifespan really quickly.
And they've got such a cute name.
They do. I do. It's a bit of a misnomer. They're actually more attracted to vinegar than to fruit, so they're more
really?
properly called vinegar flies, but fruit flies works too.
I guess when the fruit... Oh, this is total sidetrack, isn't it? I guess when the fruit starts to... it's when the fruit gets overripe, isn't it? Then that's when you start getting the fruit flies, so I guess it's the vinegar that, or some kind of sour thing.
It's everything growing on the fruit that the flies actually eat.
So they're not healthy.
Well, they're getting rid of all the stuff that's decaying the fruits.
That's
so
true.
In some ways, they're good. Yes. And they're very useful for aging research.
Yes. So they did a very good job. So you look at the fruit flies and...
Yeah, so I've been asking what happens if I change these genes just in the cells that change the most with aging, the glial cells. And it turns out when I do that, I can actually get longer-lived, healthier flies just by changing gene expression in glia. So that's another hint that glial cells really are important for brain aging, and we think also for the susceptibility of the aged brain to disease. So now what I'm trying to do, we have lots of information from different researchers all around the world, including some of my own work, who have been able to isolate these glial cells and figure out of all of the thousands and thousands of genes in the genome, which ones are changing their activity with age, just in glial cells. And some people have done this in fruit flies like me, some people have done this in mice, and some people have been able to do it from human samples, although that's a lot more challenging. But what's really interesting is it looks like many of the same genes across all of these different animals are changing in glia with age. So there's something about aging in glia that seems just universal to animal brains. But all we have now is just a list of hundreds of genes that change. So my big project at the moment is to take that list of hundreds of genes and test them one by one to see if they can protect against what we think causes Alzheimer's disease. So I'm doing that again in fruit flies because they're really short-lived, and I can actually look through these hundreds of genes and hopefully narrow it down to just the ones that are most important.
How long do you think that will take you?
Well, I currently have funding for this project from the Alzheimer's Society for 2 more years.
Right.
In fruit flies, I think I realistically can do that in that time, which is one of the great things about fruit flies. Of course, it's a far distance from actually getting to treatments in humans and in patients.
Yeah.
But hopefully I can at least identify a few genes that are worth trying in preclinical models, looking at human cells, looking to see whether there are drugs that can actually impact on that. So the broad horizon is quite a few more years down the road,
right.
But at least the fruit fly part I hope I can do in 2 years.
Wow, that's exciting.
It is.
It's amazing.
And how do you... Oh, I suppose it's just the same. I was going to say, because it's like the fruit flies are so small, and so how do you do that?
Yeah.
How do you change that? But then the neurons and the cells are small anyway, so it's like...
The neurons and cells are small, but they're the same. If you look at a neuron in a fruit fly and a neuron in a human, the size is a bit smaller in a fly, but they look broadly the same. They have the same parts of them. They fire the same way. The glial cells look very similar too. They do a lot of the same functions. So we hope that by looking at fruit flies, we're making discoveries that are actually applicable to humans as well, just on a much smaller scale. But the nice thing about flies is they have a full brain. They have neurons, they have glia that interact, we think, in the same ways they do in our own brains.
Okay.
So you're looking at this glia cell?
Yeah, glial cells.
Glia cell?
Yeah.
Yeah. Sorry.
Or glia, that's fine.
So looking at this glia and seeing whether that's, by changing them, that's what's affecting this Alzheimer's or causing Alzheimer's. Are you also, or are other people looking at why some people's glia is changing in the first place?
People definitely are. It's not something that I'm able to do in my current research, but there are a lot of people particularly looking at a variety of different diseases to see if there's any common change among glia in Parkinson's and Alzheimer's and motor neuron disease, or whether each of these diseases are really distinct and have different things changing in the glia. And there have been some really exciting studies where there are new technologies where you can take a whole brain sample, sort it out into individual cells. So you'll have thousands of neurons, thousands of glia of all sorts of different types. And then they can just look at the gene expression in each of those individual cells and hopefully figure out what's changing in people living with Alzheimer's, or people who are aging without any disease, or people who have other diseases. And hopefully that'll give us some clues. But it's all very early days with that research, but very exciting.
Very exciting. And just so life-changing.
Yeah. I mean, that's the thing. There's really right now, there's no treatment or cure for Alzheimer's that slows the progression of the disease.
No.
There are a few treatments that are able to slow the symptoms, but the disease still marches on. And I think what a lot of people don't realize is right now, Alzheimer's disease and dementia are the number one leading cause of death in the UK. They've overtaken cardiovascular disease recently.
I didn't know that.
Yeah.
I didn't know you could die from that. I sort of thought you had it and it was a part of...
So, the diagnosis or deciding cause of death involves looking at what the underlying cause is of what ultimately causes death. Mm-hmm. But right now, it is dementia, which means that this is something that's really important for people as they get older. We all know our population is getting older, and the number of people living with dementia is going to increase. So anything we can do, any treatment we can find to help slow disease progression, prevent the disease, or potentially, maybe, even cure it, would be huge. So that's ultimately what we're aiming for. So hopefully my work is helping to understand a bit better what's changing in the aging brain so we can get there hopefully soon.
So what are you doing sitting here talking to us? When does it work?
Right before this, I was in there looking at the flies. They're good for a few hours. So.
They're doing their stuff. You'll come back and check on them.
Exactly.
Very exciting.
Okay, so you're working your way through all the different ones to try and isolate down to which one it could be.
Exactly.
And that would be amazing if you could then isolate that.
So what it looks like on a day-to-day basis is what we're actually doing in the flies. Flies don't get Alzheimer's. So what we have to do is put in a protein into their genome, a gene into their genome, that makes the protein we think causes Alzheimer's disease, called amyloid beta. And what that actually does is, just like in human brains, it ends up killing off the neurons and the glial cells in the flies as well. And that protein causes them to get sick, they die sooner, their motor function is impaired, their brains don't work as well, their activity patterns are disrupted, and all of that we can see by really simple tests that we can do in the lab, that I can do with undergrads in the lab. So it's a really powerful way of looking at things really quickly. And what we can do is then tune up or tune down each of these genes that we're interested in, just in glia, in the flies, and see if it makes them better. Hopefully we'll find some that do.
Wow.
So
why can't we just take away that acid that's the protein that's in us isn't in there?
That's the multi-million pound question. And that's,
I have a lot of those.
To be honest, that's where most of the current clinical trials are focused.
Yeah.
With varying amounts of success.
Mm-hmm.
None of them so far have been approved to go through to full clinical use. But most of the strategies right now are based on reducing the amount of amyloid beta in the brain.
Right.
And I really hope that some of those are going to work.
Yeah.
But it may be that what's actually important in the brain is the reaction to amyloid beta, how the neurons and the glial cells are reacting to it, and that's what we actually need to target to.
Yeah.
So there are a lot of different things that could be going on, but hopefully one of these ways of treating it is going to work in the future.
And then sort of like maybe with that whole pincer effect, then it will actually sort of like get it under control.
Do you work with a team here?
I do, yeah. So right now in my position, I'm what's called a junior research fellow. So I have a fellowship from the Alzheimer's Society that gives me funding for my own work and for a technician to work with me. But I work really closely with the whole team of scientists in the Institute of Healthy Aging. That includes people who work on flies and worms and different animals that age more or less slowly. And we're looking at aging. We have researchers who are looking at Parkinson's, frontotemporal dementia, motor neuron disease, lots of different diseases. And hopefully, if we see things that overlap among all of our research, we can bring those together as treatments that would be useful, not just for one disease, but for many of them. But it's a great team to work with.
Brilliant. And it's so important, isn't it, that sort of collaboration, like keeping the communication so that so people know what's there and what can be used.
Absolutely.
In other areas.
And I think being at UCL is particularly good for communicating, not only here within people working on fruit flies and worms, but just across the street. We have the DRI, the Dementia Research Institute at UCL, and they have teams who are working in human cells, working with high throughput ways of looking at drugs to test if any of these drugs can actually impact on what's going on in Alzheimer's disease. So it's great to be able to take our findings, go over across the road, talk to them, and say, hey, you should be looking at these 10 genes. You should be testing drugs that work on these 10 genes. So ultimately, that's what I'm hoping that I can do. So there's the Drug Discovery Institute just across the way, the DDI, and they're working on doing exactly that, taking the results of people who work at UCL and putting them into a system that can actually identify drugs that could be used in the clinic. So it's a really exciting place to be, an exciting time to be doing this work.
Right. No, it's exciting, isn't it?
Why this podcast is, I just don't realise. I mean, I work here, we just don't know what's going on.
There's so much going on in this crowded jumble of buildings here.
Yeah.
It's amazing.
It's good. So then it's like everything... it's like real time, so they don't have to wait for you to produce your paper.
Exactly.
And then sort of like find out like
everybody,
another 5 years of, we just spent 5 years looking at the wrong 10 genes sort of thing.
And I think you've put your finger on a really important point. It takes a long time to publish findings in science. The more we can do to put out our findings ahead of publication, to communicate with other teams, to put things openly out so people can see them, the better we are as a whole group of scientists.
Yeah. And as a world.
Yeah.
So, George says, how did you get into this research? Where did you start from?
Yeah.
Interest?
So I think I've always been interested in biology. And even as a child, I went outside and played with ants and spiders in the garden, and it was something I just loved doing. But in terms of focusing it on neurodegenerative diseases, I think what got me most interested was when I was a kid. I was really lucky. I had 3 of my 4 great-grandmothers alive who I could meet when I was a kid. And I saw that the 2 of them who I spent the most time with, my great-grandmother, or Gigi Elsie and Gigi Pearl, had very different trajectories in terms of aging. Gigi Elsie spent a lot of time with me painting and playing music, and she was just completely 100% cognitively there, knew exactly what was going on. And then I also had really good times with my Gigi Pearl, but she was living with dementia, and it was a very different experience with her. For her, she'd grown up in rural Missouri at a time when there were no cars around, everybody just rode horses. So when I would go visit her, or when the family went to visit her, she would ask us, "Oh, where'd you tie up your horse?" In her mind, she was really back in a time where there were no cars. Wow. And so we got used to answering, well, out front. Where else would we tie up the horse? But I always wondered, what was it that was different about them that made old age so different for the 2 of them? So I think that was the sort of driving force to get me interested in neurodegenerative diseases. But then when I was an undergrad at Emory University in Atlanta in the US, I had a few opportunities to actually do some research firsthand, and that's really what kicked everything off. So during the term time, I volunteered in a lab that worked on fruit flies, and that got me really excited about the possibilities of fruit flies, working mostly on the genes that contribute to development, how it is that you take a bundle of cells in an embryo and then make a whole animal out of it, which was a really interesting question. But then I spent my summers doing research at the National Institutes of Health, where I was working on a disease called spinal muscular atrophy. And that really gave me an insight into how working in the lab can actually translate to, hopefully, treatments for the disease down the road, and how understanding the underlying genetics of a disease can really make a difference. Now, a number of years later, there are actually promising treatments and clinical trials for spinal muscular atrophy. So it's been amazing to see that progress, and that's really what I hope for in my own research. So that's what kicked everything off and got me really interested in doing the research. And now I get to take those 2 things together and look at neurodegenerative diseases using the power of fruit flies. So I'm really fortunate.
Playing with insects, really.
Oh,
I'm still, every day is just playing with insects in the garden. It really is. My parents look at me and say, "You haven't changed."
But wow, but you're making a difference. So that's amazing.
No, it's amazing.
Brilliant. Could you sum up why your research is so important to the public?
Yeah, I think, as I said earlier, dementia is the leading cause of death in the UK right now, and there is no disease-modifying treatment. So any advance that we can make towards having a treatment for dementia is really going to transform how the population ages, and really how healthcare looks going forward into the next 50 or 100 years as we have an older population.
Brilliant.
It's very true.
Yeah, thank you so
great
much.
work.
Thank you. Well, thank you ever so much for your time. Thank you for being here. And thank you to everybody for listening. We're going to put links up on our website, which is the antiwafflepodcast.wordpress.com, and our Twitter account, which is @antiwafflepod. Please do get in touch because we'd love to hear from you. Bye.
Bye.
Bye.